The first speaker is Dr. George Bird, who has been a nematology professor at Michigan State University since 1973. From 1991 to 93, he served as the National Director of the USDA Sustainable Agriculture Research and Education Program. And since 93, he has been a member of the Board of Directors of the Rodale Institute and has served as a member of the board of the Michigan Organic Food and Farm Alliance. Please help me welcome George.
Well, good morning. Glad to see everybody up. That was really a great tour, and thank you, David, and everybody else that wound up providing the expertise to pull that off yesterday. Well, I don't see that my first slide has come up. This morning I'm going to be talking to you about some research projects in Michigan that I've been involved in that relate to soil quality and soil health. As I was introduced basically as a nematologist during my career, I've done a number of different things. I'm going to ask them not to turn on the clock for the talk until the first slide comes up, okay, in relation to this, okay. But in the past few years, basically, I've become what I call a non-card-carrying soil biologist. And as I travel around the world, there are not an awful lot of soil biologists these days. As a matter of fact, while I'm ad-libbing here these days, I spend quite a bit of time in Central Asia, in some of the former Soviet republics, particularly Tajikistan, Uzbekistan, and Kyrgyzstan. And basically what I'm dealing with over there, which is a very daunting task, is about 3,500 years of extensive soil quality degradation and soil erosion. So basically all of us know that soil is the foundation of organic agriculture. Now I'm going into my talk. I'm going across the top row, basically, of the first slide. And sometime within the past decade, Science magazine dedicated an issue to soils. And basically they said there that soil is the next frontier for science in general, and that soil is basically a very under-researched area. When we get to that particular slide, over on the right-hand corner, you'll find another little photograph, and it's about a publication that we put out in Michigan about 5 years ago on fruit, and basically it's called Fruit Crop Ecology and Management. If you haven't seen that, it's one that I would advise you to get a copy of. We're quite proud of it. It's in a series that we call the Harwood series. And what's unique about the Harwood series is that the farmers basically told us we needed something like that. We went to work for a year. We sent our manuscript back to the growers and asked them what they thought. And they said we missed the mark. And so we went back for another year and worked, and it went quite well. Back up one for that, and let's go over it. So we've talked about the soil and the fact that we need some more scientific research there, and a copy of that publication. I'm going to be talking about observations on the biology of organic orchard soils, but I also have a quotation there. And it's from Liberty Hyde Bailey, 1916, The Principles of Fruit Growing. And it says, if the grower knows why, he or she will teach himself or herself how to make it work. And I think we saw a lot of great examples of that yesterday. If I can have the next, go ahead on that. We're going to give you an introduction. I won't spend any more time on that. And then we'll talk about 3 research projects and a summary at the end. Go ahead with the next one. Things have changed in biology because of molecular biology. We used to talk about 3 or 5 different kingdoms of life. Today, when I teach general biology, I have to teach that there are 23 kingdoms of life today that we share this planet with. And these are divided into 3 different domains. The bacteria, the archaeas, which are kind of a form of bacteria, and those with the true nucleus with the membrane, the eukaryotes. And of these 23 kingdoms of life, many of these are represented in the soil. There are only 3 that we can see with the naked eye. And today I'm going to be talking to you about 5 or 6 different kingdoms of life, and you can see that these may have some relationship to each other. Next one. We published about a decade ago the results of an experiment. And this was really our introduction to this area, where we had a conventional cherry orchard, we had cover crop, we had mulch, we had compost, and an organic system. The organic system was compromised a little bit because the farm manager didn't believe in organics and didn't always follow the instructions properly. And we had an organic consultant that we worked with there. However, you can see that we almost doubled active carbon in the organic compared with the conventional, and the same thing would be with active nitrogen in pounds per acre. Next slide. I thought it would also be beneficial for me to spend a moment on this particular diagram of nutrient mineralization. Over here I have an orchard with the root system. It's bacterialized with roots. The process of photosynthesis you can see is going on there. And basically speaking, soil organisms like nematodes, flagellates, ciliates, amoebae are very much involved in nutrient mineralization. How does this take place and why? Well, you know, the root's a little bit leaky. We'll get an amino sugar exudate, and that's fed on by the bacteria. We'll say that the average bacterium has a carbon-nitrogen ratio of 5:1. And those then are fed on by the amoeba, the ciliates, the flagellates, or the nematodes. And if I talk about the nematodes, oh, maybe a carbon-nitrogen ratio is 10:1. So basically, if you look at that and balance that equation, you get twice as much nitrogen as you need, right? 2x. So basically, the nematode mineralizes that and releases that back into the environment, right into the rhizosphere, right on the rhizoplane. Exactly the same reason you have a urinary system, to get rid of excess nitrogen that could be toxic to your body. So the two research hypotheses that I want to talk to you about today basically is the first one is that we have these organisms in the O horizon, the duff or the litter layer sitting on top of the soil, subsoil. And that's something that I was not aware of 10 years ago. Quite frequently soil scientists scrape off that area before they take their soil samples, and basically they're throwing all the good stuff, okay, away. Second hypothesis basically would be that the mineralizers in this O horizon are much more prevalent in organic systems than in conventional systems. Next slide. Well, we'll start off with nematodes, seeing I'm a nematologist. And when we compared the O horizon with 0 to 15 centimeter depth and 15 to 30 centimeter depth, you can see there was a significantly more bacterial feeding nematodes in that O horizon than in the other two soil profiles. There was no difference in relation to fungivores. I think at this time I should probably remind you that nematodes are very, very good indicators of soil quality because of their diversity of feeding habits. We have those that feed on bacteria, fungi, plants, those that feed on plants and also animals, and then the carnivores. At those three depths, there was no difference in relation to the fungivores. The herbivores, fortunately, were right where we would have predicted it, where we would have the greatest amount of root biomass. The omnivores, and I do not know why, were up there in the duff and in the litter layer. And in case I run out of time and don't get to it, I'll tell you right now something that surprised me, and I've worked with mycorrhizae for probably more than 40 years. I found many, many spores of the Endoganaceae, okay, the endomycorrhizae up there in that litter layer in the O horizon. And that really surprised me. I don't think that's ever been published anyplace. No difference in the carnivores. Next slide. So, George, you're a nematologist. What about those other organisms? So we also looked at the amoebae, the flagellates, and the ciliates, and in all cases, we found significantly greater population densities in this O horizon than at the 0 to 15 or 15 to 30 centimeter soil depth. And if you look at those p-values down at the bottom, I think those p-values are things that graduate students would probably kill for in relation to their research. Next slide. Then I guess I didn't go much into the methodology here. We went up into northern Michigan and we selected 8 cherry orchards. And these were paired cherry orchards for this particular study. And 4 of them were certified organic and 4 of them were conventional. And in all cases, the certified organic were orchards that had been transitioned. At that particular time in Michigan, I don't think we had any new organic cherry orchards. And when we separated that out, you can see that there was an order of magnitude difference, tenfold greater population densities of the, in this case I chose to show you this morning, flagellates in the organic system than in the inorganic system. So basically what I'm showing you here in relation to one group of organisms, and I don't know if that was 1 species or 10 species because I'm not an expert on flagellates, even though I see them all the time underneath the microscope. I think that particular slide is pretty good proof that there is a biological difference in the soil in commercial orchards between conventional and organic. Next slide. Systems are anything with 2 or more interactive parts. And in my opinion, all systems have 3 phases. They have a developmental phase, like getting that orchard into production. Hopefully, you're going to have a lengthy dynamic equilibrium phase. And then basically, there is a senescence, which could be a declining orchard. We saw great orchards yesterday, but I went onto my computer and came down from the satellite. And in this particular area, I could see some orchards that had an awful lot of missing trees. We didn't see those orchards yesterday, but that would be the third phase of a complex, open, nonlinear system that I don't have time to talk about this morning. Next one. I now want to sort of give you an introduction before I talk about the final experiment, but we went to a certified vegetable, organic certified vegetable production system, did a very extensive sampling there. And when we did this sampling, the place had been certified for 17 years. Basically, what we found is that 73% of the nematodes we recovered were bacterial feeders, and that 26% were fungal feeders, and a very small proportion of that being the plant parasites. But this is the number I want to talk about at the bottom. That's absolute density. Per handful of soil, there were about 3,500 nematodes. Now, about 3,000 is what you see in an awful lot of the literature for fairly active soils. But in Michigan, quite frequently, our population densities of nematodes are much, much less. So we went across the road to a conventional cornfield and did the same thing. And you can see that it was not biologically active, only about 200 nematodes there per handful of soil. Relatively few bacterial-feeding nematodes compared with the certified organic. And here, basically, with the herbivores, the plant parasites, there were 2 or 3 species at relatively high population densities for that. We also went to the adjacent woodlot. And basically, forests are relatively stable and have sort of intermediate population densities. And here you can see that we have sort of a diverse distribution of the percent of the different species in each of these categories, bacterivores, fungivores, herbivores. Old field succession, we know that's sort of in the development stage. And we've validated all this at a number of different sites, including the Kellogg Biological Station, LTER. So I just wanted to show you what I kind of call a nematode community structure analysis type of data, which I think is one way to take a look at soil quality. Some of my nematology colleagues have a much more complex system for doing that, which has some structural aspects to it and some enrichment aspects to it. But I think this is a simpler system, and I seem to be able to get fairly good results with it. Next slide. A few years back at the Horticultural Research Station at Michigan State, Clarksville Research Station at Michigan State University, we converted a typical Midwest corn soybean field into an organic orchard. And in this particular case, we relied quite heavily on cover crops between the rows, quite heavily, as you can see there, on organic mulch, organic mulching in this particular case. And what I want to show you is that it took me from a nematode analysis about 6 or 7 years to reach that dynamic equilibrium stage. So that's very much like what we were told at the last site yesterday when we were looking at that extremely healthy cherry orchard. Next slide. So let's use this nematode community structure analysis for this final data slide. My conventional standard, you see, was a very low population density, a relatively biologically inactive soil. The organic standard that I just showed you from the 17-year certified vegetable operation, very active soil with about 3,500 nematodes per handful of soil. At transition, you can see that this particular corn soybean field was fairly typical for Michigan. It was very much bacterially oriented, but overall population densities were quite low. At certification, we had really changed this. And we had sort of taken it from the bacterial organization more into a fungal orientation. But you can see that the percentage of the nematode population basically that were plant parasites had declined. Those plant parasites were both basically Predilonchus penetrans. I'm not going to have time to go into that very much. We heard a lot about Predilonchus penetrans yesterday in relation to apple replant. But remember, it's got to be that species. As a nematologist in the root lesion nematodes, Predilonchus, there are about 60 different species. Some of them are aggressive, and some of them are very wimpy. And penetrans is an aggressive one. But by the time we took the basically the straw mulch out of there, we had converted back to basically a bacterially dominated system. Our herbivore populations had gone down greatly. So you can see this is a dynamic system, and I saw changes throughout the first 5 years. In the 5th year, the 6th year, and the 7th year, I had gotten to dynamic equilibrium, which is a little bit longer than the 3-year process in the United States that's needed for certification. Next slide, please. So in summary, soil is a habitat for organisms and a habitat for nutrient fixation and for nutrient mineralization. The O horizon has the highest population density of soil microbes. Soil microbes are impacted significantly by orchard management procedures. Organic cherry orchards have significantly more soil microbes than conventionally managed orchard systems. And the soil microbes associated with the Clarksville system reached a dynamic equilibrium between the 5th and the 7th year. And the last slide, I believe, takes me back to a view at the Rodale Institute. And up there on the hill, you will see the first organic apple orchard in Pennsylvania that was ever certified. And I want to end again with this particular quotation that I believe very strongly in. If the grower knows why, he will teach him or herself how to get the job done in the orchard.